Welcome back, life science learners. Trust that you've refreshed and ready for this part of the lesson. It's important that now we look at some of the applications of understanding how DNA works. We're going to attempt to look at DNA fingerprinting. We're also going to spend some time looking at how to potentially answer a question based on DNA. It's important that we now take all of those concepts and put them together and link them to various processes such as DNA fingerprinting, writing an essay, and the applications of that to processes where mutations take place. So let's get straight into the lesson and see how this part of the segment unpacks. So the question reads, and I often remind the learners to highlight key components of the question, and we'll go through this together. Detectives were investigating a crime scene and they found blood on a broken window. They suspected that the blood was that of a criminal. To identify the criminal, they analyzed a DNA sample from the blood and compared it to that of 4 suspects. The diagram produced is on the next slide. So guys, here we're seeing what we refer to as a DNA fingerprint, and here we're seeing the sample of DNA that was extracted at the scene, and we're seeing 4 possible suspects that have been analyzed and their samples have been tested, or what we call DNA fingerprinted. And it's important that we recognize that DNA fingerprinting is a technique that has revolutionized how we can identify individuals, link them to parents, look at missing identities of individuals that have been found, deceased individuals. Also in the conservation of animals, where trade of animals such as the horn, rhino horns, we can now use that to link that to specific organisms or animals that have been poached by poachers. So it's been an amazing technique, and now let's look at how we answer a question on that. So the first question on this, name the technique that was used to identify the criminal. So guys, this technique is called the DNA fingerprint or fingerprinting, and it's essentially it's a process that we use to identify individuals. We use the rationale that no 2 individuals have the same genetic makeup, and hence when DNA fingerprinting takes place, we know that it will produce a unique band based on an individual's genetic makeup. If we see that 2 banding patterns, and I refer to these as banding patterns, which are produced by segments of DNA that have traveled through a gel that is being created, and that separates out the DNA based on their sizes. And so that separation creates what we call a DNA banding pattern. And that banding pattern will be unique to each and every individual on Earth. And that is the uniqueness that we use in differentiating individuals. Okay, so the technique is called DNA fingerprinting. Who is the possible criminal in this specific DNA fingerprinting band? So guys, the best way for us to be able to link an individual to a crime scene is to have evidence. We know that DNA can be used as evidence to link an individual to a crime scene. However, it does not conclusively prove that that individual committed the crime. So part of convicting a criminal is being able to find evidence that links him to the crime. DNA is one part of that evidence. So here we've got the sample and we've got 4 different suspects. And so I'm going to go through this and I'm going to compare each band, and that's essentially what I'd encourage you to do, is to use a ruler and place that along, and let's see. So as we look at this band, we identify that these 2 are together. So it's definitely not, there's nothing there, there's something here. There's certainly an identical match between that, and definitely Ntabi, who's got only 1, is not a perfect match. The next band, if you look at, is present there. It's present in there, it's present there, nothing there. Again, a positive match on 2 occasions. If we go on to these 2, and if we look at them together, you can clearly see that that has been similar to Jenny. If we go on to the next 2, we can clearly see that it's only found in that individual, as well as the last one. So guys, it's quite a simple process. It's looking for exact matching banding patterns. This wasn't particularly difficult for us to be able to identify. And the best way for us to be able to do that is to run a ruler down and tick off, and at the end you'll generate a band pattern that has exactly a match of that. So in this case, we've identified the individual, and the individual is Jenny, and you can clearly see how that banding pattern is a 100% match of that individual sample that was found. Okay, explain your answer to question 2, and that's important. For 2 marks, you must be able to explain the rationale for why you came to the conclusion that it was that specific individual. So you've got to explain yourself correctly and appropriately using the correct terms. So when we look at Jenny's DNA profile bands, there's a match or a perfect match with the DNA profile bands taken from the crime scene. If you look at it, it means that because there's a perfect match, it means that these 2 samples have to have come from the same individual. And in this case, we know that the sample of DNA that was found is a match to Jenny. And so that question is, now we've got to identify what was Jenny's reason for being there, or her reason, the reason for the blood having been on the window pane. And those are, again, information that we need to further investigate at a crime scene. State the next question. State one other use of this technique identified in question 1. So the technique is DNA profiling. As I mentioned, it's used firstly in solving crimes. And that's the principle based on identifying individuals and finding matching DNA samples to that individual. That's kind of the most convenient and easiest answer that we can use. But apart from that, we know that DNA fingerprinting can be used to prove paternity. Now guys, paternity again means relatedness to a set of parents. We often see questions in an exam where it references babies that were mixed up at the time of birth, and so we need to then use the DNA of 4 possible parents, a set of parents, to link a child to each of those parents, and that's working out paternity, so the parents. We also know that individuals that go missing and often can be established. So there's lots of human trafficking around the world that happens, and so it can link, DNA sampling can link individuals to a set of parents as well as to an individual that has DNA being stored in a DNA databank. The identification of genetic disorders. So another technique where DNA fingerprinting works is the ability to be able to screen individuals and look for genetic disorders in their genetic makeup. And that's a fantastic discovery that has led to becoming more clinical in the treatment that we administer to people. Establishing family relations. So again, in terms of siblings as well as individuals related to parents, we know that siblings would have some percentage of match of DNA. In relation to the parents, you'll find that that complement of DNA that you and I have is made up of genetic information from both parents. So we can establish relatedness by looking at DNA fingerprinting. Matching tissues for organ transplants. Guys, one of the challenges of organ transplants is that there is often what we refer to as tissue incompatibility. And that means the tissues often from different individuals are not compatible. One of the screening methods in terms of looking at compatibility between a donor and a recipient is to do a genetic fingerprint. And that again looks at typing the genetic information to see how similar their genetic profiles could be, and that often reduces the risk of rejection of organs during transplants. And then lastly, identifying dead persons, animals, etc. So as I mentioned earlier on, when a suspect has been found with, for example, a rhino horn, in South Africa, we're able to there's a project that has fingerprinted all rhinos around in the country. And so that allows an individual that has or was found in possession with the rhino horn to be able to track us to a specific rhino and where that rhino is from. So that can then link an individual to a crime scene. So it's important that the law now has evidence to be able to link individuals to a specific crime scene. It's also done for the trade of some of the exotic plant species like cycads, which are often traded on an international market. And so this can be used in terms of genetic fingerprinting plants as well. So that's an interesting question. As I mentioned, it's important that we recognize that there are essays in paper 1 and paper 2 that often can include sections on DNA, RNA, and protein synthesis. So let's look at one specific question and see how we would answer that question. So the question reads, describe the location and structure of DNA, the process of DNA replication, as well as the significance of the process of mitosis. So guys, as we unpack the question, it's important that we recognize what are the different parts of this question. And this helps in planning. And I often remind you that in writing an essay, it's important that we read the question well. We identify the different segments or parts of the question, and that we brainstorm in a plan before we get down to writing the essay. This all requires time, but if done well, it ensures that you're able to comprehensively answer your question when you have to pen down your ideas for that question. So let's look at how I would go about doing that. Remember that the content is 17 marks. We have a 3-mark allocation for the synthesis, and I'll reference what synthesis is later on when we look at the answers. And it's key to understand that there are no marks awarded for answers in the form of tables, flowcharts, or diagrams. So if you decided that you learnt the process of, in this case, DNA replication, it will be good if you use that diagram to help you explain your answer. Unfortunately, you will not be given the mark for having a diagram that illustrates the process of replication. So yes, it's important to use diagrams to understand the process, but in the essay, it's important that we look at how content is synthesized into, in this case, the process of DNA replication. So, as I've illustrated on the next slide, I've had to take the question and unpack that into the 3 different components. In this case, the first part of the essay described the process of DNA replication. The other part needs us to describe the location and structure of DNA. So we're going to spend some time allocated to the location and structure of DNA in our essay. And finally, describe the significance of the process of mitosis. So guys, it's important that we recognize that in any of your papers that several concepts can be linked together in an essay. And that's essentially how we're able to synthesize and string together, in this case, a response to the question. So in your answer, you've got to start off with describing the location and the structure of DNA. So where DNA is found, describe what are the building blocks of DNA, double helical, the nucleotides, the nitrogenous bases, and the complementary base pairs. I'll go through a suggested answer for that. We also then need to look at the process of DNA replication. Again, if I were to break up the 17 marks, I would simply allocate 7 marks to this, probably 5 to that, and then another 5 to that. That's just my understanding before even attempting the question. So if I'm able to get 5 significantly important facts for the description of the location of the structure of the DNA, and then another 5 for describing the importance of the process of mitosis, I would have had a kind of a clear overview now of what the mark allocation would be, so that when I write them, I note that at least 5 points and another 5 there, and possibly 7 for the process of replication will give me a total of 17. Okay, so in terms of describing the location, it's important that we recognize that part of the question required us to describe the location. We know that DNA is found in the nucleus as well as the mitochondria, and make special emphasis of the chloroplast in plant cells as an organelle that contains DNA. Then in terms of discussing the structure of DNA, we've got to be able to describe any of these 7 facts. So DNA is double-stranded. It basically is a helical molecule. It's made up of monomers called nucleotides. Each nucleotide consists of 3 parts, a sugar, a phosphate, and a nitrogenous bases. And then systematically discuss the 4 different types of nitrogenous bases. And finally, include the concept of complementary base pairing and how they are held together with hydrogen bonds. So if you think about it, it's quite systematic in terms of the way you can arrange your answer, and think about it logically. Think of the diagram, double-stranded helical structure. Then you discuss the nucleotides, and you also mention the base pairs and how they're bonded together. Okay. Also part of the question required us to be able to explain the process of DNA replication. And if you look at the mark allocation to this, was 6 marks. And so again, if you are traditionally inclined to being able to have that image of DNA replication in your head, it makes it easier for you to be able to string together this process. Again, guys, remember that this is only my suggested memo. We're expecting you to be able to write in a congruent flow of ideas and concepts. So please do not just bullet point them, because that's not what is expected in the essay. It needs to be ideas and concepts that are well strung together. So the DNA, you've got to describe it unwinding. You've got to talk about unzipping and the hydrogen bonds breaking. This then causes the strands to separate out, forming two templates. These templates then serve as strands on which the free nucleotides or the complementary DNA nucleotides will bond, where adenine to thymine and guanine to cytosine base pair. And this is using the free nucleotides in the nucleoplasm. This results eventually at the end in the production of two new identical DNA molecules, each molecule consisting of one original strand and one new strand. And that's essentially the overview of the process of DNA replication. Remember that the third part of the question was discuss the significance of DNA replication for mitosis. And guys, we know that mitosis was a process in which the cell copies and produces two identical cells. And we know that during interphase, we know that DNA replication occurs. So in this bit, you've got to be able to quickly describe that genetic material is double-stranded and that each cell receives the same amount of DNA, and it's important that the DNA replication takes place to ensure that the daughter cells have a genetically identical copy of that information. And finally, the synthesis of this essay is based on the three criteria: looking at the relevance of your facts, looking at the logical sequence of how concepts are linked together, and how comprehensive you are in terms of putting together your information. So often learners tend to forget about these three important marks, and that's crucially important in terms of knowing that there's no irrelevant information, that your information is logical and sequential, that you're describing processes like protein synthesis or DNA replication correctly and logically, and then finally, being able to, in this case, the significance of a process such as mitosis. So guys, that's a wrap for this lesson. We've looked at an overview of all the concepts around DNA and the code of life. We've looked at various different questions on how we apply our understanding. I trust that you've enjoyed the lesson. Please go and attempt some questions. Take out those textbooks, look at the revision activities in them, download some papers, attempt these questions, and I hope that you improve your understanding. From my side, cheers, have a baie lekker day.